Hello, everyone, and welcome to this session titled Leukodystrophy 101. This session is intended for newly diagnosed families or people who are interested in learning more about what leukodystrophies are as a disease family. My name is Norm Berberick, and I'm the Global Patient Advocacy Lead for Rare Diseases at Takeda Pharmaceuticals, a proud sponsor of this conference. It's my pleasure to introduce our speaker. Dr. Paul Watkins is a research scientist at the Kennedy Krieger Institute and professor emeritus at the Johns Hopkins University School of Medicine, where he has held joint appointments in the departments of neurology and biological chemistry. A primary goal of Dr. Watkins' research work in leukodystrophy is to understand the biochemical basis of X-linked adrenoleukodystrophy, or XALD, and related genetic diseases. This work includes identifying drugs that lower very long-chain fatty acids in cells from ALD patients, and then testing them in mice with ALD. Research in the Watkins lab has also focused on a family of enzymes involved in the metabolism of fatty acids called acyl-CoA synthetases. Although now retired, Dr. Watkins continues his research and consults on ALD. Please join me in thanking Dr. Watkins for his research in ALD and his contributions to the leukodystrophy community. Dr. Watkins, we look forward to your presentation.
Well, thank you very much, Norm, for those very kind words. And so if we can put the slides up on the screen, I will be happy to get started. Are we good to go? I don't see the slide up on the screen. If the moderator could let me know.
Yes, we can see your slides up on our screen.
Excellent, excellent. Okay, well, this is Leukodystrophy 101, and it's supposed to provide some fundamental information about what is leukodystrophy and how it applies to all of you. So leukodystrophy comes from the Greek, and if you break it down, leukos means white, dys means ill, and troph means nourishment or growth. So if you put it all together, something about problems with growth and nourishment of something that is white that causes an illness. If you look at dictionary.com, you'll find the definition: any of several inherited diseases characterized by progressive degeneration of myelin in the brain, spinal cord, and peripheral nerves. Degeneration of the white matter of the brain characterized by demyelination and glial reaction, probably related to defects of lipid metabolism. Don't worry about these words yet. We're going to come back and dissect these definitions in a minute. The ULF website defines leukodystrophy by saying the 50+ leukodystrophies make up a group of rare genetic disorders that affect the central nervous system by disrupting the growth or maintenance of the myelin sheath, which insulates nerve cells. These disorders are progressive, meaning that they tend to worsen throughout the life of the patient. And then if you look at the information on the NIH National Institute of Neurologic Disease and Stroke website, and I truncated this from an overly long group of paragraphs. Leukodystrophy refers to genetic diseases that predominantly affect the white matter of the central nervous system, or CNS. Leukodystrophy is not a single disorder. It is a group of rare, primarily inherited neurological disorders known as the leukodystrophies that result from the abnormal production, processing, or development of myelin and other components of CNS white matter. Leukodystrophies are usually progressive, meaning they get worse as time goes on. Some forms are present at birth, while others may not produce symptoms until a child becomes a toddler. A few leukodystrophies mostly affect adults. So we see some common themes in all these definitions: brain and central nervous system, white matter, myelin and the myelin sheath and demyelination, inherited, genetic, growth and development of myelin, lipids and lipid metabolism, and progressive and degeneration. So let's look at a little more deeply into what leukodystrophy is with these items in mind. So the nervous system can be divided into the central nervous system, or CNS, and the peripheral nervous system, or PNS. The central nervous system consists of the brain and the spinal cord, whereas the peripheral nervous system is all the nerves that emanate from the spinal cord and go out to your hands and feet and legs and all of the organs in your body. And I apologize for the ringing telephone. I have no control over that landline noise. I'm sorry. It'll stop in a few seconds. So if you look at an anatomical specimens of the human brain, this you see from a side view on top here, where the front is to the right, and this is the frontal lobe, and the back or occipital lobe is to the left. The cerebellum is underneath the brain, and you can see a little bit of the spinal cord and brainstem underneath the brain. If you look at the top down, this is what the brain looks like. It's not smooth, it's irregular, and has these valleys and that increase the surface area of the organ. If you take a slice through a section of through the human brain at this level, shown through the cartoon skull here, you'll see that this is what it looks like when sliced. You'll note that there are areas around the periphery that are pinkish or grayish, and then the area in the middle that is white. So this is the gray matter and the white matter of the brain. So when we're talking about white matter diseases, we're talking about diseases that affect the material that appears white in this brain section. If you take another slice, and this is sliced from top to bottom of the brain, from left to right, for example, if you did it at the level of the ears, you would see, again, a lot of gray matter in the periphery and white matter in the center. And the same with a horizontal slice, similar to the one we showed on the previous slide. Now you'll see also there are holes, or they're not really holes, but they are vacuoles. These are the lakes and rivers in which the cerebrospinal fluid percolates through the brain. There are stains that will stain myelin, and that stains the white matter of the brain, as you can see in this coronal and horizontal section. So all of this appeared white in the unstained brain and appears bluish, dark bluish to black in the stained brain. So this is the white matter. Here's an MRI view that many of you should be familiar with. And in a brain of a patient who is not affected by leukodystrophy, you see that in contrast to the anatomical specimens, the white matter looks dark in an MRI T2 weighted image, which is a more common MRI that you have probably seen. The ventricles, or the lakes and rivers that the cerebrospinal fluid percolates through, are white on a normal MRI. So let's look a little bit further into the brain. The brain, like all other organs and tissues, is made up of billions and billions of cells. And although there are many types of cells in the brain, there are four main types. There are neurons, or nerve cells, oligodendrocytes, which make myelin, astrocytes, which help form the blood-brain barrier, and microglia, which is like a mini immune system in the brain. And all of these, except for neurons, are referred to as glia, or glial cells. We'll talk about each one of these in a little bit more depth in the next few slides. So neurons, which are the cells that carry electrical impulses from one place to another, look like this cartoon view here. There is what we call the cell body, which is the main part of the cell which contains the nucleus. And then you see these little processes coming from the cell body that we call dendrites. And then you see a long process coming from the cell body, which we refer to as the axon. And the axon is what carries electrical impulses from the cell body to the next cell that is connected to the neuron via a synapse. Synapse is just the point of connection between the neuron's axon and another cell. Now, this other cell could be another neuron, could be a muscle cell, and so on. And as you can see up here, the dendrites of this neuron are getting connections from other neurons shown by these bluish lines. So these would be axons from other neurons. So there's a vast interconnected network that's formed by synapses between axons and dendrites. Here's another view of it, and this time showing that the axon is covered with what looks like insulation, and that is the myelin. We'll get more into more detail about myelin in a few slides. As a matter of fact, in the next slide, we'll start with that, because the next cell type, called oligodendrocytes, are the cells that make or synthesize myelin. And as you can see in this cartoon, here's the cell body of an oligodendrocyte. And here is all this membrane that is being it's part of the oligodendrocyte's cell membrane, but it wraps around the axon from this neuron and helps to insulate the wire. Here's another cartoon view of it showing that one oligodendrocyte can produce myelin that covers more than one neuron. And it's further illustrated in this scanning electron micrograph of oligodendrocytes, showing that again, a vast network of membranes produced by the oligodendrocyte that help to form the myelin sheath around many, many axons of neurons. The next cell type is astrocytes, and they help form what we call the blood-brain barrier. It helps protect the brain from unwanted materials, but it also creates a barrier between the bloodstream of the brain, which is in these mainly small capillaries, small blood vessels we call capillaries. As you can see, the astrocyte, and here's a better picture of a real astrocyte stained with a green fluorescent dye. They are very branched, and these little things we call foot processes completely surround the external surface of the blood supply to the brain and allow nutrients to come into the astrocyte, which the astrocyte then passes on to the neurons. So the astrocyte is critical for nutrient delivery, like sugars, from the blood to the neurons. Then we end up with the fourth type of typical brain cells called microglia. And these microglia have immune system functions. So in this picture to the right, the green cells are astrocytes, the red cells are microglia. And the microglia just are kind of hang out as sentries. And if something, for instance, toxic or a bacteria happens to get into the brain, these microglia are on patrol to help to eliminate any unwanted material that enters the brain. So let's look at a couple of cartoons showing the brain cells all in one context. So here we have two neurons, the cell bodies up here. You see that there are lots of axons that are sending or that are making synapses with the cell body of these neurons, but these neurons themselves are sending out a couple of large axons that are surrounded by myelin being made by the bluish oligodendrocytes. The astrocyte, only one of which is shown here in green, these have their little foot processes all over the blood vessel, and I'll show you a more accurate representation in the next slide. But they're making connections between the blood supply and the neurons. In this cartoon, we see a couple, in this case, mainly a neuron here and here, excuse me, the blue here and here, which are sending out axons, and they are covered by myelin, which is being made by oligodendrocytes. There's an astrocyte, and as you can see in this, that's a neuron. This is these orangish cells are the astrocytes. As you can see, the processes at the end of the astrocytes completely cover the capillaries, making it impermeable to anything that the astrocyte does not want to let in, and let it be permeable to the things the astrocytes do let in. And then the astrocytes can transfer that material to the neurons. And so you see in this upper part where the cell bodies of the neurons are located, this is the gray matter. And in the lower part where you have myelinated axons, this is the white matter. And this just happens to be a cartoon of a section of spinal cord, but it's the same for the brain. Both the brain and the spinal cord have gray matter and white matter. And so when you have a neuron whose axon is myelinated, it's not a continuous sheet, as you can see, that it is interrupted by spaces that we call nodes of RNVA. And those nodes actually help to make the signal travel more efficiently. And as you can see here in this blow up, this is a cartoon of how the myelin is wrapped around the axon. Here's an electron microscope view of an axon with myelin surrounding it. And this is very typical. We'll show a couple more of these views in a minute. Right now, as a matter of fact, myelin development begins in about the 3rd trimester of pregnancy and rapidly accelerates after birth. It is most rapid in the first 2 years of life. But as you can see in a newborn, it's pretty much low in myelin compared to an adult, where the very dark stained material or myelin sheaths. We continue to myelinate throughout life, but it's most active in the first 2 years of life. So one last view of oligodendrocytes, which I thought was an interesting analogy of what the myelin sheath looks like. If you can imagine a window blind, where when the window is open, you see the myelin sheath stretched out, but when the blind is rolled up, you see a view like this. And this is very similar to what myelin looks like. So what does myelin do? What's its function? The main function seems to be insulation of nerve cell axons to prevent short circuits. You don't want your wires crossed. If you cross your wires, you'll get incorrect signals and cause lots of problems. And the discontinuous nature of myelin with those nodes of RNVA, it actually increases the speed of electrical impulses. What's myelin made of? Well, it's about 20% protein and 80% fat, which we call lipid. The myelin proteins include 4 that are listed here. It includes more, but these are 4 major proteins found in myelin. One is called myelin basic protein. One is called myelin associated glycoprotein, or MAG, myelin oligodendrocyte glycoprotein, or MOG, and proteolipid protein called PLP. I'll show you a picture of some of them in a minute. Myelin lipids include, and these are names that some of you may have heard in the context of your particular leukodystrophy of interest, galactocerebrosides, galactosylceramide, sulfatide, sphingomyelin. But there are many more lipids in fatty substances that make up myelin, but these are some of the main ones. How about myelin proteins? Well, if you take a look at this cartoon neuron and axon with myelin around it, and look at a blow-up view of the myelin here, the surface of the myelin is on this side, the neuron is on this side, and as you can see, MAG and MOG are mainly found on the surface of the myelin sheath. Myelin basic protein, on the other hand, is found more toward the interior near the neurons. But interestingly, the PLP, proteolipid protein, you find several places, and the PLP from this membrane interacts with the PLP from this membrane and kind of acts as an anchor so that to keep the myelin, rolled up myelin from unrolling, so to speak. You don't have to be concerned about anything in the box in the context of this discussion. How about the myelin lipids? So lipids are fats. What makes fat fat? Well, chemically, long chains of carbon atoms and hydrogen atoms, and the black ones are the carbon atoms and the white ones are the hydrogen atoms. They're the essential features that make fat fat. As you know, oil and water don't mix, and these are very oily substances. And because of that, because it excludes water, that makes a good insulator because it does not conduct electricity very well compared to more water-based substances. So leukodystrophy, many leukodystrophies are characterized by demyelination. What do we mean by that? Well, let's suppose we have a couple of neurons here, the cell bodies are up here, and the axons are going this left to right, and they're covered by myelin. What happens if, let's say, a toxin or a virus, let's say a virus comes along, let's say a virus that somehow demyelinates or destroys the myelin in certain parts of the certain neurons, you get degradation of the myelin and you get naked axons. At that point, what happens? Well, two possibilities are, if the virus is contained and eliminated, you can have remyelination, where oligodendrocytes come in and make more myelin and put things back to normal. So here would be the starting point with myelinated axons. In the middle, we would have demyelination, where at least some of the myelin has been destroyed. But if this continues, and there is further degradation of the myelin, it can lead to even further degradation of the axons and cause this picture in the middle. On the other hand, if you get remyelination is illustrated with the electron microscope picture on the right side. So in leukodystrophy, if the problem that causes the demyelination does not get resolved because there is a genetic mutation in protein or so on that will cause the demyelination, if there is no cure for that, things get worse, and this is part of the progressive nature of leukodystrophies. Again, many leukodystrophies are characterized by demyelination, and if you look at a section of brain or nerve, you'll see a picture similar to this, where there may be some myelin remnants, but it's far less normal appearing than in the view on the left side. Demyelination can also be shown in slices of brain. In an unaffected brain, the myelin will stain blue with a stain called Luxol fast blue. Whereas in this section from a patient with a leukodystrophy, you see very little blue staining. So, what are the possible etiologies of leukodystrophy? There's a genetic possibility, an environmental, for example, a toxin like PCBs or so. You can have an infectious insult like a virus or a bacterium, or you can have immune system dysfunction. For the purposes of this talk, we're going to focus mainly on the genetic causes, genetic etiology of leukodystrophy, because that's what we see primarily. So by genetic cause of etiology, we mean a mutation in a gene. And these mutations can either be inherited from parents, or they can be new mutations, which we also sometimes call de novo mutation or spontaneous mutations, where a child has a leukodystrophy and has a mutation in a gene that they did not inherit from the parents. But most leukodystrophies are inherited, which means that gene mutation comes from one or both of the parents. Talking about genetics, there are several modes of inheritance that we know of. There's recessive inheritance, which we also refer to as autosomal recessive. There's X chromosome linked inheritance, and there's dominant or autosomal dominant inheritance. As you probably know, humans have 23 pairs of chromosomes. They have 22 pairs of what we refer to as autosomes, and there are 2 sex chromosomes. And as I'm sure most of you know already, that women have 2 X chromosomes, where men have 1 X chromosome and 1 Y chromosome. And so when we talk about autosomal recessive inheritance, we're talking about something going on with one of these autosomes, whereas X-linked inheritance refers to the sex chromosomes. So most leukodystrophies that we know of are inherited in an autosomal recessive manner. So what does this mean? So in this cartoon here, the dominant gene, or the one without a mutation, is in green, and the recessive gene, or the one with a mutation, is in pink. And so with an autosomally recessive inherited this trait, such as a leukodystrophy, the father has 1 dominant and 1 recessive copy of the gene, and the mother has 1 recessive and dominant copy of the gene. Now, if the dominant gene from the father and the dominant gene of the mother are passed on to the child, you have a child who is doubly dominant and would be unaffected with the particular disorder or leukodystrophy. On the other hand, if the recessive gene from both mother and the father is passed on to a child, you have a child that is affected by the disorder. Also, you can have a dominant from the father and recessive from the mother, or dominant from the mother and recessive from the father, in which case you will have 2 offspring that are essentially like the parents. They have a dominant and a recessive copy of the gene, but most of the time, they don't show any symptoms of the disease because the dominant one is sufficiently strong enough to overcome any deleterious effects of the recessive gene. Only when you get 2 copies of the recessive gene do you wind up with clinical disease. Some leukodystrophies are also inherited in an X chromosome-linked manner, as shown here. So in this case, we're showing that the father has a normal X chromosome and Y chromosome, a dominant in both cases. And this case, the recessive gene, the gene with a mutation, is also carried on an X chromosome, one of which the mother has, and the mother typically also has another dominant or unaffected gene. And so the way these can be passed on is if the X chromosome, dominant X chromosome of the mother plus the dominant X chromosome of the father get passed on, since it's XX, it will be an unaffected daughter. If the dominant X and the Y from the father are passed on, it will be an unaffected son. If you pass on the dominant dominant X from the father and the recessive X from mom, it'll be just like mom. She'll have an unaffected daughter, but who will be carrying the recessive trait on one of her X chromosomes. And the problem lies where the recessive X from mom and a Y chromosome, which is by definition dominant from the father, get passed on. We have a son who has a recessive X chromosome, and since there's not another X chromosome to balance it, this individual will be affected by the disorder. So let's look at how rare or common is leukodystrophy. So some leukodystrophies are not all that rare. X-linked adrenoleukodystrophy has a gene frequency of 1 in 17,000 in the population, in the worldly population. Even more common, in 1 in 40 people of Ashkenazi Jewish ancestry have a mutation in the gene that causes Canavan disease. But many leukodystrophies are rare, or at least they seem rare. And what needs to be answered is this due to just the generally infrequency of this particular gene mutation, or is it underdiagnosis? And that's things that clinicians and epidemiologists are very interested in understanding. But if we look at the overall incidence, all leukodystrophies, it's approximately 1 in 7,500 births worldwide. So when do we suspect that a child or an adult has a leukodystrophy? Well, one thing is gradual decline in an infant or child who previously appeared well. Progressive loss in body tone, movements, gait, speech, ability to eat, vision, hearing, and behavior are often seen, as well as slowdown in mental and physical development. But symptoms may vary according to the specific type of leukodystrophy. And very importantly, they may be very difficult to recognize in the early stages of the disease. In adolescents and adults, the rate of disease progression can be slower and less severe than in infancy and childhood. Movement disorders such as movement disorders as well as seizures and dementia are common in this age group. So how about clinical diagnosis of leukodystrophy? So very important is the history of the illness, the clinical progression that observed mainly by parents for young children. Neurologic examination is of course very important, and the presence or absence of non-neurologic symptoms always has to be considered. And family ethnic background can shed some light, as some disorders are more common in certain ethnic populations than others. Not all, but some. Brain MRI, of course, is one of the key diagnostic tools that neurologists use to diagnose leukodystrophy. So let's look at some brain MRIs for a minute. In this slide, we're looking at an T2 weighted MRI scan of an unaffected brain. And as we mentioned before, myelin, or the white matter of the brain, appears dark in this form of MRI image. In leukodystrophy, and this is an extreme example, but nonetheless,
Sarah, just for 5 more minutes.
illustrated
I
that
know, but you can juggle like I usually
the
do.
white appearing material that you do not see in the unaffected brain is evidence of leukodystrophy.
I'm gonna sit on this couch with you because you don't.
Early MRI changes are often subtle, and the MRI abnormalities generally worsen as the disease progresses. MRI changes can be unique to different leukodystrophies. While there are some commonalities, there are some patterns that emerge, as you can see, looking at MRIs from 8 different types of leukodystrophy. Neurologists and radiologists are trained to look for patterns, and pattern recognition on MRI can often suggest a diagnosis, a specific diagnosis that can be verified by other testing. Other testing includes biochemical testing, and by which we mean laboratory tests. And these are used along with clinical findings and MRI to confirm or refute a diagnosis. The biochemical tests are generally specific for a single leukodystrophy or a group of related leukodystrophies, but sometimes panels or groups of tests for the more common leukodystrophies can be ordered by the doctor and can be very useful in sorting out the issue in an undiagnosed case. But no single test can generally answer the question, does the patient have a leukodystrophy or not? So how many different types of leukodystrophy are there? Well, the ULF website lists 56 clinically distinguishable leukodystrophies, but a review of the medical literature suggests that there are more than 100 different leukodystrophies. So genetic defects in leukodystrophy can result in biochemical abnormalities. And so let's look at some of these things. You can have accumulation of a toxic substance, and we'll get into that in a little more detail in a minute. You can have mutation in a specific brain protein, or you can even have too much of a specific brain protein that can cause problems. So let's look at toxic substances that accumulate in specific leukodystrophies. In X-linked adrenoleukodystrophy, there's accumulation of very long chain fatty acids. In MLD, accumulation of sulfatide. In Krabbe disease, galactosylceramide accumulates, and in Canavan, N-acetyl aspartate is the substance that accumulates. Interestingly, these three, the first three are lipids, but the fourth one is a different type of chemical that's not a lipid. And by toxic chemicals, what we're talking about are things that are normally present in the brain, but the balance between just enough and too little or too much has been disrupted. It's the Goldilocks problem. So using the seesaw as an analogy, on one side of the seesaw, you have the amount you take in of a substance and the amount you make of a substance. And on the other side, you have the amount you excrete, either in the urine or feces, for example, or the amount that you break down or degrade by normal processes. And there's a balance that needs to be achieved by these processes. When you have a toxic compound, there's two likely causes of excess. The first is that the body cannot remove the excess, in which case the breakdown is not working properly. And so you get too much because intake, synthesis, and excretion are not effective. Or the body just makes too much, where synthesis is increased and intake, excretion, and breakdown are normal. So these are the typical causes of some of these toxic compounds. And where do they cause problems in the brain? Well, we said that sulfatide, MLD, galactosylceramide, CRAB-A, very long chain fatty acids, FA, are lipids, and they cause problems with the myelin sheath. In contrast, the N-acetyl aspartate in Canavan disease is a problem with the oligodendrocytes. Another biochemical cause of leukodystrophy is mutation in a specific brain protein. For example, GFAP, or glial fibrillary acidic protein, is a problem in Alexander disease. And in Pelletier-Merzbacher disease, the myelin protein called PLP, or proteolipid protein, is the problem. And like some of the other lipids that we've seen, and you know that from earlier discussion that proteolipid protein is found in the myelin, this is the location of the issue in Pelletier-Merzbacher disease, whereas GFAP in Alexander disease is mainly an issue with the astrocytes. So we're going to kind of end up with genetic diagnosis of leukodystrophy. As you all, I'm sure, know, DNA sequencing is very important in diagnosis of leukodystrophy. And by DNA sequencing, we mean the order of A's and C's and G's and T's in the patient's DNA. There are different subcategories of DNA sequence, including sequencing of a specific gene. For example, if the clinical picture suggests that a young boy has ALD, we can just sequence the ALD gene and see if there are mutations in his ALD gene that are consistent with a pathological consequence. Next is exome sequencing. DNA contains both sequences that code for proteins that have the blueprint for making proteins, or sequences that do not code for proteins. And if you just sequence the parts of the DNA that code for protein, that's called exome sequencing, and this can be very valuable in establishing a diagnosis. Finally, the expensive alternative, but the most complete, is total DNA sequence via genome sequencing. And this can be extremely valuable, particularly in establishing a diagnosis for a family with an undiagnosed leukodystrophy. Recent publications indicate that a combination of clinical findings, MRI pattern, and genome sequencing can dramatically shorten the time from initial symptoms to definitive diagnosis. It's not uncommon to wait 10 years between the initial symptoms and a final diagnosis in the past, and hopefully this is getting shorter and shorter and shorter. Dr. Vandiver, who I believe is attending this ULF meeting, came up with a diagnostic algorithm for diagnosis of leukodystrophy that was published in 2020 in the Annals of Neurology. And it's instructive to go through this, because I think this shows you the process that the doctors go through to try and figure out what's going on. If you have an abnormal MRI and you think that it's likely a genetic etiology, you check that out. And if no, it's not a genetic abnormality, it's likely to be acquired, like an infection or trauma, and you would treat it. You would treat the infection or the trauma. But if yes, it's likely genetic etiology. Does it have a recognizable MRI pattern? Well, if it does, then you pursue confirmatory testing and confirm the diagnosis. And if that gets confirmed, then treat according to the standards of care and return the results with the appropriate counseling to the family. But if the MRI pattern is not recognizable, then you need to proceed to some biochemical testing or DNA sequencing. And if these prove to be correct or prove to suggest a diagnosis, then you confirm it and treat accordingly. Or if the whole genome sequencing suggests something, assess to see whether there are P for pathogenic or likely pathogenic mutations in a likely disease-related gene. And if that is in fact the case, then you confirm the diagnosis by other methods and treat accordingly. And if it's not confirmable by orthogonal assessment, unfortunately, we're left with referring the family to rare disease advocacy, research programs, and so forth. Organizations like the ULF are well geared toward helping families who unfortunately do not achieve a specific diagnosis, either before or after genome sequencing. So what are the approaches to treatment for leukodystrophy? Well, the holy grail is remyelination and a restoration of normal brain function. That's the ultimate goal. So possible therapeutic options include, for some leukodystrophies, enzyme replacement therapy; for some leukodystrophies, bone marrow transplantation, sometimes also called hematopoietic stem cell transplantation. Neural stem cell transplantation is, I know, on the horizon, but I don't know currently what the status is. Gene therapy is, of course, of great interest, with the corrected version of the gene being delivered either by a virus, lenti or adeno-associated virus. There's also antisense oligonucleotide therapy, and which I'm sure a lot of you have heard great things about, is CRISPR-Cas gene editing. This is a technique which, under ideal conditions, can actually go in and correct the mutation in the specific gene and literally correct it. I'm not sure what the current status of CRISPR-Cas is with any of the leukodystrophies, but this is something that's on the horizon. And do not neglect supportive care. Supportive care is the mainstay of treatment until we have a diagnosis and a specific treatment. So in summary, leukodystrophy refers to a group of diseases affecting the white matter of the brain. White matter is primarily myelin, the fatty insulation around nerve axons or the wires. Degeneration of white matter is due to problems with growth or development of myelin. Most of the leukodystrophies are inherited or genetic diseases. Most are unfortunately progressive, and some still remain undiagnosed. But identifying causes of undiagnosed leukodystrophies is getting faster thanks to whole genome sequencing, and progress toward developing effective therapies for some leukodystrophies is very encouraging. So I thank you for your attention. I apologize that I could not be there in person this year, hopefully next year. And if you have any questions, I'll be happy to try and answer them. And my email is on the first slide if anyone has further questions that we didn't get to here. So thank you.
Questions?
So how does the leukodystrophy is different from like Alzheimer's and Parkinson's, and what's happening in the brain?
Unfortunately, I am a biochemist, not a neurologist, and I don't have a good hard answer, good scientifically founded answer to your question. I do know that you mentioned, did you mention Alzheimer's and Parkinson's? Is that correct?
Yes.
Yes. So Alzheimer's, we are beginning to learn that there are possibly different gene etiologies for Alzheimer's disease, and there's not one specific gene. Like we know with many of the leukodystrophies, there's a very specific gene that carries a mutation that causes the disease. But dementias, including Alzheimer's, are what we call multifactorial, and there could be many genes that could contribute to the progression of the disease. And there are specific toxic substances that we do see in the brains of Alzheimer's patients. With respect to Parkinson's, there is a specific set of neurons that are in the brainstem that are that degrade for some reason. And so I believe, I could be wrong, but I believe that Parkinson is more of a neuronal disease rather than a leukodystrophy. So I'm sorry if that's a poor answer, but you really could probably get a better answer from a neurologist.
Any other questions?
There is a question that came in the chat.
It doesn't appear there are other questions. Thank you. Oh, hold
Well,
on.
I have one in the chat. Maybe it's a silly question. Can you hear me okay?
Yes, and there's no such thing as a silly question.
Well, I'm just curious if you have any advice on either how to slow down the progression of the demyelination or how to help regrow the white matter, the myelin sheath.
Do I know of anything? Not specifically, but what I do know is there are many research groups who are very interested in both slowing down the progression of demyelination and or restoring the myelin. Remyelination is, as I said on that one slide, is really the holy grail of treatment. You know, if we can figure out how to trigger remyelination, or alternatively, if we can, for example, if it's a leukodystrophy where there's the buildup of a toxic substance, you know, if we can halt the buildup of the toxic substance and clear out the excess, there are natural pathways for myelination that can kick in and hopefully remyelinate. Myelin synthesis does not stop in childhood. Myelin synthesis continues throughout adulthood. So if we can figure out how to control what's causing the demyelination, the remyelination mechanisms may be able to sufficiently kick in and help restore some normalcy. Again, there's an incredible amount of research going on in both of those areas. And there's no such thing as a silly question. It just means that it's a question that other people in the room were thinking of, but were too scared to ask.
Well, thank you. I'm a teacher, so, you know, I use that philosophy as well oftentimes. But what were the websites? Do you know off the top of your head any kinds of websites, or maybe it's somewhere right in front of my face where I may be able to research how to recreate sort of the myelination?
Unfortunately, I do not on the top of my head have any of those things. And I think that if you start talking to some of the neurologists who are physically present at the ULF meeting, you might be able to pick their brains. That's what they're there for. They're there for you. So don't hesitate in approaching anybody at the meeting who might be able to help you. If you listen to other talks and hear what their special areas are, they may be able to help you. I would suggest Dr. Fatimi or Dr. Vanderknap, just off the top of my head, but they're Dr. Vandiver. But there are probably more than a dozen excellent people at the meeting who could give you a better answer than this one.
Okay, thank you.
I have just a comment on that discussion. Someone was talking to me recently about remyelination in zebrafish. So if you're looking for websites, I recommend a Google for zebrafish and remyelination. I think for MS is why they were studying that.
Zebrafish?
Yeah.
Yes, zebrafish. Zebrafish are a good model for certain leukodystrophies, it turns out.
Interesting. I will. Thank you.
You're welcome.
All right, any other questions? If not, thank you, Dr. Watkins. We really appreciate your spending this time with us.
Thank you. It's a pleasure to be there. Sorry, next year I'll be there in person. Hopefully can meet some of you.
Yeah, me too.